Hello. Thank you for giving me this opportunity. My name is Yutaka Akiyama. We are from Tokyo Institute of Technology, Japan. I'm a computer scientist in training and have long been working in the area of bioinformatics. It was only 3 years ago we started our small activity to somehow help drug discovery for NTDs. Today, I will briefly introduce our open resource called iNTRODB and some experience in its application. It was year 2012. We initiated a non-profit research consortium in Japan. At this moment, we have virtually no funding, yet the members are very experts in this field, and we can combine our expertise very well. The members include 3 universities, Tokyo Tech, ourselves, and the University of Tokyo, and the Nagasaki University, Institute of Tropical Medicine, as well as 2 national laboratories, KEK and AIST. KEG provides a huge particle accelerator and beam lines which can be used for the X-ray crystallography. And one of the most important member is Astellas Pharmaceutical Company. They are very strong at structure-based drug design. They have their own beam line on KEG. And they have a very quick pipeline for getting structure information. The role of Tokyo Tech computing teams, 2 things. The first 3, target protein selection based on bioinformatics techniques. And in second, virtual screening using our supercomputer environment. But today I will focus on the first one. When we started this activity, we did not set any priority among 3 trypanosomiosis. However, currently we are trying to focus on the Chagas disease with gentle advice from the NDI. But very recently we found we are facing a kind of impact of this policy change. Maybe I will discuss later. In the workflow of structure-based drug design, as all of you know, we need a good protein target at first. Such a target protein should have a physiological relationship with the target disease, and structure information is crucially important in SBDD, of course, and hopefully we would like to have in vivo and in vitro experimental information about that protein. However, the issues, well-known issues in drug discovery for NTDs is the relative shortage of knowledge if compared with the major human disease like cancer. For example, we do not understand the gene regulatory networks and metabolic pathways in pathogen of each stage of parasite life cycle. And we do not understand the interaction between the host metabolite, I mean human cell metabolite with these parasites and so on. So naturally, our strategy is to utilize the available information for the other homologous species. Here, we must mention that there are several excellent existing works like a TRIP-DB and the TDR target. However, in this particular target, our requirement was slightly different. In order to strongly support SBDD approach, we needed some special functions. Because we can determine the 3D structure by ourselves, we need to evaluate the feasibility of structure determination of the target protein, as well as the binding pocket information from Campbell, available or not, and so on and so on. So we have decided to create our own small new tool to help our decision making in SBDD. That is INTRODB. INTRODB stands for Integrated Neglected Tropical Disease Database. And it integrates the information from PDB, structure database, Campbell, GeneDB, and metabolic pathway, and so on. Using INTRODB, user can make a target protein search. Typically, the user starts from choosing a genome. In this illustration, it's a trypanosome brucei genome selected. So the system shows you the listing of the whole protein sequence registered. And each sequence information is linked, hyperlinked to the resources inside or outside. For example, PDB entry with remote or cross homologies, and the Campbell experimental information of the cross homology sequences, KEGG pathway information, and other genome information to see the conservation. And these related information are shown in a single line on the INTRODB display. And just by clicking these cells, user can reach to the hyperlinked information. The web interface for INTRODB are freely available on the internet. Okay, let me show you the one more example. In this case, we started from Cruzi genome. There are 10,000 proteins registered, and the 1,000 of them have some PDB structure information with the sequence similarity threshold of 0.001. And among them, 600 has some related Campbell information. And if you wish to do so, you can avoid conservation with a mammal protein. In that case, the number decreases to the 110. So after focusing on the small number of proteins, the user can further investigate the characteristic of each target proteins. Just for example, the user can see, just by clicking this button, user can see the pathway information. And on top of the pathway diagram, we put also the RIT-seq information. But this is for the brucei experiment. And in this example, the target user chosen is colored in red. It means a lethal protein in brucei. 3 years ago, in our actual project, we started from brucei genome sequences and started from 9,000 proteins. We put these selection conditions, meaning lethality and very strong conservation among 3 trypanosome meiosis, and very close structures available in PDB, very tight condition, I think. We selected 117 candidates at first. But 100 proteins are too big number for us, so after that we made manual inspections, and by the discussions with experts, we chose 4 targets. These 4 targets were selected mainly by the availability of the 3D structure and the feasibility of our own structure determination. One of them chosen is or was spermidine synthase. This enzyme is important in polyamine biosynthesis, and it's highly conserved with 3 trypanosome, and the low with Homo sapiens. And SRM is crucially important, lethal in the RIT-seq experiment for Cruzi. So we believed this is one of the good target. For these 4 first targets, we performed a virtual screening bioassay, and Astellas pharmaceutical team made several co-crystallization. And in vivo assay was also performed in Nagasaki and STPH. And the good news is that for these targets, we have found several micromolar or sub-micromolar weak hits with a variety of scaffold. It's a good news, but bad news. One of the bad news is that the SRM inhibitor, which worked very good in vivo for Cruzi, but did not work at all for Cruzi in vivo. I do not know why, but membrane permeability seems to be enough, so I don't know, but maybe some relationship with the host metabolite or something. Okay, maybe it's time to stop. And today I would like to introduce our small activity in Japan. We have a collaboration work with a consortium, non-profit consortium, and the iNTRODB is an open resource. Thank you so much for your kind attention.